metabolism, excretion, toxicity. I had enough of a window to know that I wasn't going to have to go through all of this, so that's what it all is. But the bottom line is that together, this is what describes the disposition of the compound in vivo. The animal is not a test tube. So the second issue, and we've heard some of this today, limitations of translational models. And I will speak exclusively in the realm of neurological disease, psychiatric disease too, CNS disease. The poor predictive power of CNS animal models for efficacy of candidate therapeutics in humans has been recognized as one of the major reasons for the high attrition rate of CNS drugs in the clinic. We have limited but growing knowledge of pathogenesis in neurological and psychiatric disease, and we've developed animal models for behavioral and pathological aspects of these diseases. They're pharmacodynamic models of aspects of the diseases that we're trying to treat. We test exploratory compounds and candidate drugs in these animals, and we cure them with a significant success rate, the pharmacodynamic outcomes that we're trying to treat. But we have not done an adequate job of aligning the models with the stages of the human disease that are being investigated in clinical trials. So the translational value of these studies has been largely inadequate. And I'm going to obviously fill in the picture here. So that's just the beginning argument, preclinical, clinical. So abundant new knowledge has accumulated over recent years. The rhetorical question, are we incorporating these developments into our translational strategies effectively? The answer, of course, is no. What have we actually been testing in preclinical animal studies? Do our translational models inform only prevention trials? And I'm going to make an argument that in many cases the answer to that is yes. Is there a preconceived bias in how we use our models that actually disconnects them from effective translation to clinical studies of patients enrolled in trials? I would argue the answer to that question is yes. How can we refine our models to make them more informative in clinical applications? And conversely, how must clinical trials be modified if existing translational models are to be informative? So here's a paper that was published in Nature Reviews 2 years ago this month. First author was Giovanni Frassoni, and let me just orient you to this because it's going to come up in the next few slides. It's an example. It's just an example. I live in the world of Alzheimer's disease, so I'm using Alzheimer's disease as an example so I can be comfortable up here. I did it again. Good. This is the asymptomatic phase of the disease. It's been recognized that the disease actually begins pathologically and probably in reality a good 10 to 20 years before there are any clinical symptoms. So this is the asymptomatic phase. This is what we're referring to as mild cognitive impairment, or MCI. It's when, beyond the initial memory problems that the worried well may be experiencing. This is the first thing which is characterized. And then there's dementia. And this aligns neuropathology with that. So in terms of cognitive markers, this is where we first see memory test changes. Okay? There's rapid maximal impairment, and this is useful for diagnosis of MCI. But it plateaus, and it has limited usefulness for tracking disease progression from MCI into dementia. Verbal comprehension changes start later. So these are indicative of the progressive dementia, but they are of limited use diagnostically in the early stages of the disease. So they're more sensitive at the dementia stage. We take the same figure and look at the natural progression of pathological markers, and there's some play in these curves. There's been some refinement. But by and large, this still is the picture that's accepted in the field. Amyloid markers, and these are measured by CSF analytes, by PET imaging, amyloid PET imaging. These are detected early, in advance of symptoms, but they plateau by MCI. Some would argue that that means amyloid's not involved in the disease. Others would argue that that means it's a pre-symptomatic harbinger of the disease and identifies patients at risk. Who's right? We'll see. We don't know yet. I have my biases. Functional and metabolic markers, this is functional MRI, FDG PET, These are abnormal by MCI and actually track pretty well through MCI and progress into dementia stages. They're not useful as general diagnostic biomarkers. So you're not going to be doing FDG PET and functional MRI to diagnose patients who are coming into memory clinics, especially when you don't have a disease-modifying therapeutic to treat them with. Structural changes, structural MRI, follow the temporal pattern of the clinical disease, brain atrophy, and tau pathology. So what does this all lead us to? What are the strategic impacts that this knowledge should have? Well, for example, there are many, but I would argue, and others, this is now being published in mainstream literature, that anti-amyloid therapies will likely work only as preventatives, and that administration following the onset of dementia up here, when other neurodegenerative processes have taken over, will likely be mild to non-existent. There are other processes, when you're in this stage, you're losing neurons, you're losing synapses, and if you want to engage the disease at those stages, those are the targets you're going to have to be focusing on. So if you want to look at amyloid as a therapeutic target, it's back in this stage that you have to do it. Well, that's a problem, because how do you identify the patients? So the keys is identification of patients at risk in their prodromal stages, and there is ongoing work in that regard. Understanding where existing translational models are relevant. If we're curing amyloid deposition in mice, that's cool, that's great. But that translates to this part of the human disease, not this part where we're enrolling mild to moderate AD patients in our clinical trials. So we need to align our preclinical studies with the corresponding stages of the disease progression. What do we need to do? We need to capitalize on the growing knowledge of pathogenesis in human disease. We need to develop the technical capabilities to enable translational studies, and I mean truly translational studies, between animal models and human disease. And we need to ensure that the new knowledge from the preclinical animal models that hopefully are as well characterized as what we just heard about for ALS mice, and we know that that isn't the case in AD, are applied at the appropriate stages in human clinical trials. So example, CSF, I'm just going to throw up some examples here. These are papers that were out. I cheated for this presentation. I didn't revise these next few slides, so these papers are all a year old or more. But these are CSF biomarkers in human subjects. We're getting really good at identifying the temporal pattern of CSF biomarkers in humans. But we're not applying this sort of knowledge to our preclinical models effectively. And why? Well, there's good reasons for that. It's hard. It's hard to get CSF out of a mouse. It's hard to get CSF out of a rat. But it's hard. Is it as hard as a failed phase 3 clinical trial? Image-based biomarkers in humans and disease models, there's growing recognition that neurological and psychiatric diseases are disorders of neural networks, not simply single molecular entities, single drug, single target. They're network disorders, and disorders of overlapping networks that have hubs where they intersect. The brain as a three-dimensional integrated circuit. These are circuit disorders. So here's examples. Default mode network activity, cortical hubs, impaired default network function, regions of interest, and we're getting this in humans. These are cutting-edge studies. But we're not doing it so much in mice. Here's an example of one such study in the TG2576 mouse. This is not commonly done yet. And there's a crying need for this. So we're beginning to apply these sorts of approaches in models, and these efforts need to be expanded. Again, to align the animal models with the human disease. So potential. This is an example, an emerging field. And we haven't heard anybody talk about this today. But if you go to the SFN meeting, Society for Neuroscience, 2 years ago, there was one row of posters on optogenetics. And you couldn't get to the posters. They were 30 people deep. And 2 years ago, last year, there were rows of posters on optogenetics. And you couldn't get to the posters because they were 30 people deep. This is exciting stuff. And it's emerging. It's not there yet, but it's getting there. So channelrhodopsin and halorhodopsin are genes that are capable of turning neurons on or off in response to diverse colors of light. And I apologize for anybody who knows more about this than I do, because my knowledge of all of this is secondary. But they can be selectively expressed using the Cre recombinase and define cell types, and this affords transgenic models for optogenetic control. So you can illuminate axons far away from the opsin transduced cell bodies, and this enables viewing projection-based control of cells and elucidating neural network connections. And this has been taken to primates with viral-mediated transduction. So this is a seminal paper by one of the original developers of this technology, Carl Dieseroth, January last year. I'm not going to read the whole paragraph, but just to highlight some of it. Owing to technological limitations in probing intact neural circuits with cellular precision, our current understanding of brain disorders does not do full justice to the brain as a high-speed cellular circuit. Rather than conceptualizing the brain as a mix of neurotransmitters, ideally we would be able to move toward a circuit engineering approach. All right? So this is an emerging field. This is something, and anybody who wants to get in touch with me to get these references, they're all PDFs sitting on my hard drive. Just send me a mail. So, examples, methodological developments, and I'll just run through this quickly. You know, these are methodological developments of the channelrhodopsins, the halorhodopsins, and this is undergoing very active refinement now. There's gene engineering on these opsins that are really tightening up the wavelength of light to which they respond, their on time, off time, so that there can be greater precision brought to turning on these neural networks, turning them off. This is a very active area of research, and there were some pretty exciting talks in that regard at last year's neuroscience meeting. Optogenetics and neurological disease, again, this is a year old, okay? We have three papers here where this has been brought to studies of Parkinson's. Psychiatric disease, antidepressants, cocaine conditioning, anxiety, circuitry of addiction, and then schizophrenia. This is growing. How do we align the preclinical studies in animal models with human disease? We have to take two focus approaches. One is on the animal studies. The other is on the human studies. For the animal studies, we need a better development of imaging and fluid biomarkers. We can't just keep doing the things the way we've been doing them. We need a better understanding of the neural networks that are affected in the human disease and how the corresponding networks are affected in the mice. Do the mice use the same network to conduct their executive functions that we lose when we get demented? We need the ability to push the models to a point that corresponds to later phases of the clinical disease, so we're not just doing prevention studies in the animals. And use these measures as markers and as surrogates. On the human studies, we need a better understanding and identification of prodromal presymptomatic disease that'll make the animal models more useful for translational purposes. And face it, somebody comes into the clinic and half of his temporal lobe is blown out in MCI, you may be able to treat that individual with a good new drug. It's going to be a blockbuster, but that's not going to be nearly as good as identifying the patient presymptomatically and preventing the onset of the dementia. So optimal is development of preventive therapeutic studies. There are 4 such initiatives underway to my knowledge that are focusing on genetic and biomarker-based identification of presymptomatic patients that are currently under consideration for NIH funding and other funding agencies, multinational initiatives, 3 of them based in the US, 1 of them based in Quebec. And these are going to be taking therapeutic agents they began as natural history studies of patients who had genetic mutations with a predictable time of onset of symptomatic disease, and looking at family members who carry the mutations or who don't as the controls, and looking at the biomarkers, the brain imaging, the CSF analytes, 5, 10, 15, 20, 25 years in advance of the onset of dementia, and then seeing how that progresses. So natural progression studies, and there are plans to bring therapeutic agents into these patients 10 to 15 years before the onset of dementia. And I think that is where we have the greatest hope in, at least in the remaining time of my career, and many of you in the room, of actually coming up with a therapeutic that's going to be relevant to treat Alzheimer's disease and stem the tide that's going to destroy our medical care systems financially. So third, and this will be my last issue, and I was happy to see this come up in some of the discussion today from the floor. Is single target, single drug still a viable strategy? Or have we begun to exhaust that within the pharmaceutical industry, which is where I lived for about 90% of my career? There's an emerging field called quantitative and systems pharmacology. This combines computational and experimental methods to elucidate, validate, and apply pharmacologic concepts to the development of small molecules and biologics. The goal is to understand in a precise and predictive manner how drugs modulate cellular networks. Now, I'm not talking about neural networks. I'm talking about metabolic networks, pathways within cells, within tissues, to impact on human pathophysiology. And it spans biochemical, genetic, animal, and clinical approaches and moves beyond single drug, single target interactions to a quantitative understanding of drug action across all levels. Pharmacokinetics, pharmacodynamics, and toxicokinetics at a system-wide level. Drugs disrupt homeostasis. We have extremely well-refined homeostatic controls. When one system goes down, another system steps up to compensate for that. And the same thing happens in the brain, which is why the neurodegeneration proceeds to such an extreme point before we see patients with Alzheimer's disease, with Parkinson's disease. That's why the disease is already well advanced by the time the clinical symptoms are manifest. It's important to recognize toxicokinetics and pharmacokinetics can have very different temporal vectors in various organs exposed to a drug. You want to deliver a drug to the brain, you get it into the brain, great. You know, take it to your executive team. But it's also going to the liver and the spleen and the pancreas. And it may have off-target activities in those other organs that don't have the same potency and the same PK/PD as it does in the brain. But over time, when we're talking about an elderly, frail population on polypharmacy, these are going to have effects that we're ignoring, but we can't. All right? They can have very different temporal vectors in various organs exposed to the drug. And exposures and outcomes vary among target organs, genotypes, lifestyles. Does this person go out and walk and do aerobic activity every day? Or does he sit and eat potato chips in front of the Super Bowl? Or both? So examples, and I mentioned this earlier, on-target toxicity that led to the post-marketing failure of rofecoxib, that's Vioxx, and has hindered the development of glitazones, is because of activities that the drugs have in other target organs. And they were predictable. So quantitative and systems pharmacology, QSP, will require elaboration of predictive pharmacodynamic biomarkers that assay directly the effects of drugs in cells, in tissues, in preclinical models, and in patients in order to understand the outcomes of disrupting system homeostasis. No drug acts in isolation at only a single target. The ultimate outcomes, if this is implemented, I'm painting a really hard picture here, but these are the ultimate outcomes. It's going to accelerate drug discovery and development by helping to identify druggable networks and reduce efforts on networks that are not druggable. It will facilitate the design of better drugs and drug combinations, repurposing, and the selection of appropriate doses and dosing regimens. It will facilitate the identification of patients who are most likely to respond to new therapeutic agents and combinations. We have to stop thinking, example, of Alzheimer's disease as a homogeneous disorder. It's a heterogeneous collection of syndromes that converge on a similar clinical manifestation, But that doesn't mean that we treat them all the same. We have to identify who's going to respond to what therapy at what time in the course of the disease and be able to identify those patients. And highly recommended reading, which you can't see here. But this was a white paper by the QSP workshop group constituted by the NINDS last October. I thank Susanna Pedancesca for turning me on to this and making me an evangelist. I will share this with anybody who asks me. I have it as a link, I have it as a PDF. Send me an email if you need my contact information, call me later, talk to the organizers of the meeting, just ask for it, and I'll send it along. Very highly recommended reading. This is a philosophy that needs to spread within the drug discovery field. So the take-home messages. In animals, it's easier to establish dosing, PK/PD, ADMET, toxicokinetics. This should be the translational challenge in appropriately staged models once we accept how the models fit within the disease continuum in order to inform controlled clinical trials with defined enrollment criteria. Then comes the challenge of moving from control. I'm assuming everybody in the room who's doing drug discovery really is doing it because they want to treat patients and not just to have an IND approved or to get a phase 3 trial that's going to get authorized by the regulatory authorities. We want to treat patients, right? Then comes the challenge of moving from controlled trials into population-based clinical treatments of patients with comorbidities. That's where the rubber hits the road. This is all complex, it's expensive, it's time-consuming, particularly when integrating QSP into the equation, but it's less so than ongoing clinical trial failures. And Howard, don't ask me how this is going to get funded. And I would argue that we need to embrace the challenges rather than continue with business as usual in order to improve the success rate for developing effective therapeutics and to move beyond the low-hanging fruit, which is what the industry has delivered to date and isn't delivering with the same consistency. That's all. Thank you.
Thank you for a great talk and for highlighting the QSP white paper. Just to give credit where credit is due, the white paper is the product of the National Institute of General Medical Sciences.
I meant that. I said NINDS, didn't I?
Right.
NIGMS, I'm sorry. Yeah.
And is a result of several years of workshop activities that they have held on systems biology and its marriage to systems pharmacology, and this is the product of those discussions.
Thanks for that correction. So before Hugo comes to the microphone, one of the big points in that white paper is the importance of doing failure analyses on clinical trials, and I was delighted when you brought that to the floor earlier today.
So how do you propose, in the context of you sort of warned us to think about the talk before, so sort of marrying the two, how do you think QSP is going to affect how we think about developing animal models and the robustness with which those experiments need to be conducted?
I don't think QSP necessarily helps us to develop animal models, but I think animal models will help us flesh out the systems biology that we're impacting with therapeutic compounds. And I think we are going to need a collection of well-characterized animal models in order to be able to do that effectively. We need to understand what's happening physiologically at the systems level in the animal, and then be able to understand how that relates to the similar systems biology that's going on in the human patients that we're treating.
So maybe to clarify, what I meant is, if you go into a systems level, you're obviously changing, you're increasing the number of variables, potentially leading to the output. So the question is, even with a very robustly developed model, I'm wondering whether there would be a premium on the N, if you will, on what needs to be actually tested.
You're not increasing any variables. I want to make that clear. You're increasing no variables. You're getting your arms around the variables that exist and are being ignored.
One challenge, of course, if you talk about systems biology and circuits, is going from, let's say, the molecular target, like a beta amyloid intervention or a tau intervention, to a functional outcome, which actually is what you want to measure in the human patient population. You want actually to have a functional outcome there. So what do you think about the combination of symptomatic treatment and disease-modifying treatment? Is that the kind of way forward to combine the two, or?
It's what's happening anyway. It's unethical to require patients in Western societies to take a disease-modifying drug as a true placebo control without a symptomatic drug on board.
No,
Unless they don't.
I mean, internal drug discovery, trying to go after a multi-target drug who
That's-
combines both
that's-
symptomatic and disease-modifying aspects.
That's a good question. So, I would have to posit back. What are we altering in the animal models if we're putting them on a cholinesterase inhibitor, and how does that compare to what we're altering in humans when we put them on a cholinesterase inhibitor? We need the physiologic outputs to be able to understand what the impact is, because you're not going to have the behavioral outputs in order to be able to compare. It's a problem because we're talking about soft subjective endpoints that relate to executive function and quality of life that you can't expect to model in an animal. So you need something which is physiologically monitoring and predictive and quantitative.
Various speakers have described what I could call a selectivity problem, which is a lack of specificity of drug action, typically from the perspective that drugs always, in effect, have more than one target response. Your point was really, really interesting, that even if you had a perfect response of only one target in the body, there's going to be a compensatory response, which will upregulate to compensate, and that upregulation will probably activate a variety of genes and produce a variety of proteins. So you'll actually get a rather broad systemic effect, side effects, without even activating more than one target.
Thank you.
Hi, I have a question. So the graph that you showed of the progression of disease up to that...
Idealized graph.
Right,
Yeah.
up to that first stage when it barely becomes detectable symptomatically. Does that mean that if somebody comes up with, let's say, biomarkers for the earlier stages, that should become, let's say, a part of a physical starting at a certain age? Like, for example, the only thing that comes to my mind is that, people should start getting colonoscopies after 50. So should people start getting screened for something after a certain age to see if they're in that first part of the curve?
I'm amused by your choice of analogy, colonoscopy, because that actually has been used. I was at an ADNI executive meeting, Alzheimer's Disease Neuroimaging Initiative, where there was a discussion about the resistance societally about having lumbar punctures. You're certainly not going to get a spinal tap when you go in for your annual physical, but they can get guys to go in and get colonoscopies after they're 50, which is 3 days beginning to end and pretty invasive. But a spinal tap, if you don't have the headache that results from it as a side effect, which is it's there, it's a problem. It's a few hours and you're done. What we need is better pre-symptomatic diagnostics and non-invasive, inexpensive. I'm thinking back to Dr. Weiner's, I forget your first name, I'm terrible with names, presentation on what the idealized drug would look like. Well, I could come up with a similar slide for an idealized diagnostic. All I can say is I'm working on it with my team, but we need that. We're not going to be able to do population-based screening with brain imaging and with spinal taps. But if we can improve that to a point where you can actually do with a physical or biochemical fluid-based biomarker, identify patients who are on that vector in this stage of the disease, then that should become part of general population health. And then, of course, cascades, because there's nothing we can treat them with right now, except, excuse me, get out there and get aerobic exercise and get mental activity and adjust your diet, lifestyle things. We can do that now. But we can enroll those individuals into clinical trials for disease-modifying agents, and then hopefully, we may have disease-modifying agents already that will work in this stage of the disease, but we haven't tested them in those patients yet.
Right, that's what I meant, that would make certain animal models more relevant.
Yes, because the animal models that we have developed. All right, I shouldn't just say yes as a firm statement. It's speculation, it's hypothesis based on this natural history study of human disease that the animal models we have developed to date for Alzheimer's disease are sitting around this part of the natural history curve. But that's not validated.
I'm just curious, if you get the ideal biomarker for Alzheimer's disease prevention, what kind of regulatory hurdles are you going to have from the FDA to get these really early onset prevention 10, 20, 30 years ahead of time?
It's a really good question, but what I can say is that this is exactly the question that the Alzheimer Prevention Initiative, the Dominantly Inherited Alzheimer Network, the A4 initiative that's being started up by the ADCS, they're talking about therapeutic trials on asymptomatic patients on the basis of biomarkers and genetic outcomes. And they are engaged with the regulatory agencies on that. I don't have a better answer for that question. I don't work for the FDA. But I think there is an acceptance that this is something which is going to have to happen, and it's all going to come down to the safety profile of the therapeutic compounds. There are other ethical issues too. So, for example, the API initiative, which is based on, is focused on that Antioquia family in Colombia, in the Medellin area. This is the largest family through which a particular presenilin mutation runs, and they have 1,300 family members who are registered with this trial, and a potential 3,000. And their grant application was submitted to NIA last June. The decision is outstanding. They have identified, to my knowledge, a therapeutic agent, which I don't know, that they want to bring into this population. The Colombian regulatory authorities said, so, you want to take 650 asymptomatic patients and treat them with this therapeutic compound, and they're all Colombian? I don't think so. So to spread the risk, they're now looking to incorporate sites in North America which aren't going to have the same exact genotype, but there will be familial mutations that are part of that. So there's a cascade of effects. There's the regulatory, there's the ethical. Got to get research ethics board. We call them REBs in Canada. Investigational research boards on board here too, because you're going to be treating asymptomatic patients with a therapeutic regimen that is going to have a safety profile.
So, thanks for that great discussion. In terms of the answer, the FDA is very interested, from our conversations with them, in today seeing things added as secondary arms to studies, phase 1 and phase 2. The greatest problem right now, because there are technologies which SOPs are evolving that are systems biology based, metabolomics based. It's very difficult to get funding for it out of academia because there's not a good program in NIH to fund these. And so that's really, it's investigator-initiated research in the domain of human biology. So even if your local committee approves it. So it's observational, it's looking for landmarks, and then you would actually work backwards towards the animal model from it. Because one of the problems actually in using some of these minimally invasive studies, systems biology, is the gray-white matter of the rodent versus the human. So you're going to have to actually very carefully pick, I forgot the expression you used, but it's the idea that you do have networks, we view the brain as being like a pituitary gonadal axis, let's say, only it's within the brain.
Integrated circuit.
Integrated
regions of
circuits,
interest,
yeah. So the players in the integrated circuit's a little different between the rodent and the human. So you'll have to also.
But there are
deal with
some
that.
similarities. There are some
Sure,
overlap.
there are tremendous overlaps,
Yeah.
but you just as far as the quantitative endpoints, you're going to have to deal with that, but it's doable. But right now, I think actually for academia, it's really a funding problem.
Mhm. Hand off to Diana.
Hello, everyone. So for those of you I haven't met yet, I'm Diana Shinneman. I'm Assistant Director for Scientific Affairs for the Alzheimer's Drug Discovery Foundation. I just want to thank all the speakers from last night and today for some excellent talks, and to thank all of you for attending. It's been really had some great discussions so far. We have a record number of attendees this year, so we're excited this conference is growing, and please give us your feedback as you go through the today and tomorrow as we continue to revise this conference each year. It is an annual conference. So I just have a few quick announcements. I know everyone is anxious to get to the networking reception. I think Howard mentioned yesterday during his talk that we're going to be webcasting all of the presentations. So if anyone's been taking crazy notes, you don't have to worry. We will have all these presentations available to you to access online, and we'll send the link out for that when it's ready. We're also working on a drug discovery tutorial that'll be a web-based platform, and we're hoping to have this live in the next few months. So this will be an additional lasting educational tool from this conference. So at this time, I just want to announce our Young Investigator Scholarship and Award winners. These scholarships recognize the early achievements of talented young investigators and seek to encourage their career development in the field of drug discovery for neurodegenerative diseases. All of the winners receive free conference registration and the opportunity to present a poster.